Hello, my name is Molly Matkovich, and I am a member of the Buchanan lab, and today I will be presenting role of subclinical seizures in sleep-wake regulation of a mouse model of tauopathy. Alzheimer's disease is a neurodegenerative condition characterized by progressive worsening of dementia symptoms. While memory loss is the most recognized trait of Alzheimer's, patients often experience sleep disturbances and subtle epileptiform activity, which is only observable when the electrical activity of the brain is being measured via electroencephalography. Seizures without outward signs are called subclinical. The mechanisms underlying these seizures in patients with Alzheimer's are little understood, but they could complicate disease progression. A pathological hallmark of Alzheimer's and other dementias is the aggregation of microtubule-associated protein tau. Hyperphosphorylated tau may form aggregates of insoluble protein known as neurofibrillary tangles. These neurofibrillary tangles are also present in brain tissue from patients with temporal lobe epilepsy. This leads to the hypothesis that tau pathology leads to subclinical seizures, which contribute to cognitive impairment and sleep-wake disruption in patients with Alzheimer's. It is known that the PS19 mouse model develops tau neurofibrillary tangles over time, comparable to the way in which humans do. Therefore, these mice are used as an animal model for tauopathies. 10 of these PS19 mice and 10 of their wild-type littermates were implanted with EEG/EMG recording devices around 9 months of age. Then, recordings were taken over a 24-hour period while they were between 10 and 11 months of age. These recordings were analyzed to find the number of spike wave discharges, as well as the sleep-wake phase in which the discharges occurred. Figure 1 is an example of a subclinical seizure spike wave discharge in one of the PS19 mice. Looking at Figure 2, it was found that PS19 transgenic mice had more subclinical seizure spike wave discharges than did their wild-type littermates, who do not express the P301S mutation, which promotes tau aggregation. Figure 2a shows an increase in average seizures per hour over 24 hours along the Zeitgeber 12 light hours, 12 dark hours timescale. Figure 2b expresses the number of spike wave discharges occurring in both light and dark. Figure 2c shows that for the PS19 transgenic mice, these spike wave discharges tended to happen during the wake phase, while the wild-type animals had a more even spread. Also, the wild-type mice showed discharges during rapid eye movement sleep, while PS19 transgenic mice did not. The discharges experienced by PS19 transgenic mice were between 1 and 2 seconds on average. The wild-type mice showed discharges which were a bit longer on average. The lack of spike wave discharges during rapid eye movement sleep for PS19 transgenic mice implies sleep disturbance. In Figure 3, a further breakdown of the mice's sleep-wake phases are provided. As shown in Figures 3a through 3d, PS19 transgenic mice experienced increased wakefulness, seemingly caused by increased average wake bouts. This could suggest the transgenic animals had trouble going to sleep, hence more time spent awake. Figures 3e through 3h show a decrease in time spent in non-rapid eye movement sleep for PS19 transgenic mice compared to wild-type littermates, yet an increased number of non-rapid eye movement sleep bouts. Also, the bout length of non-rapid eye movement sleep is lower for the PS19 transgenic mice. This could suggest the PS19 transgenic animals are having more fragmented sleep. Figures 3i through 3l show the same trends for rapid eye movement sleep bouts, once more suggesting transgenic animals are experiencing less prolonged sleep. To summarize, the increased spike wave discharges experienced by PS19 transgenic mice were coupled with an increase in sleep disturbances. This could affect their disease progression due to the deleterious effects of lack of sleep on mental state. The sleep the PS19 transgenic mice did achieve was more fragmented. Figure 4 shows the transitions between sleep and wake states. One can see overall from Figure 4a, there were less transitions from wake to non-rapid eye movement sleep, suggesting the animals were having trouble going to sleep. In the future, we plan to repeat these experiments on mice of different ages, ideally from 3, 6, and 9 months of age, so that we may see how increased amount of tau aggregation affects sleep and the number of spike wave discharges. Also, attempting to decrease the number of spike wave discharges experienced by a sample of mice could yield interesting data, as it would suggest how decreasing seizures may positively or negatively affect the sleep experienced by the animals. This could be achieved by administering anti-epileptic medications. With that, thank you for listening to my presentation, and thanks to all who have enabled and encouraged my research experience.